So yes, I am Stefano MARZI, and today I will speak about how RNA modification will impact the host pathogen interaction. So we are in the lab. I am working in Strasbourg in a laboratory that is actually studying RNA regulation in bacterial adaptation. But our favorite organism, it is Staphylococcus aureus. That is a major human pathogen. And today I will not only speak about Staphylococcus aureus, but I will show you in different pathogenic bacteria how RNA modification are sometimes very important in order to allow the interaction with the host and the adaptation to the different stresses that pathogenic bacteria encounter when we'll have to deal with the host, with the immune system, and with other stresses due to the host. Just a few words. In Strasbourg, I am working in this institute, the IBMC, and our unit is a whole unit of the CNRS that is dedicated to the study of RNA regulation of gene expression in different organisms. So there are two main topics in the unit. One, it is really better deciphering the decoding rules across evolution. So we are studying translation and the ribosome, the tRNA, and the messenger RNA. And the other topics that is important in our unit, it is understanding the roles and the functions of regulatory RNAs. So regulatory RNAs can be the microRNA in eukaryotes or the small regulatory RNA in bacteria, or even the structure of messenger RNA that have some regulatory function. Okay, so as I told you, we are going to speak about bacteria. So bacteria, they are really remarkable organisms because they are able to adapt to different environments. So they are able to adapt to high temperature, low temperature, and different pH range, saline conditions. So they have the capacity to change their metabolism and to change their proteome to reflect the new environment in which they are living. And this is very important when a pathogenic bacteria or an opportunistic pathogenic bacteria, for example, has to colonize the host, that can be an animal or human host. And we know, for example, that in our body there are more bacterial cells than human cells. And sometimes they are commensal, sometimes they can become pathogenic. So this bacteria has to face the immune system. This bacteria has to face different stress conditions, as we are going to see briefly. How they are doing that? It's very important the concept of stress. So the first thing, the bacteria has to sense the stress, has to understand that something has changed, and then need to reprogram his proteome in order to get used to this new condition. So bacteria are indeed stressed, but they have figured out different mechanisms how to cope with this stress condition. And which kind of stress can be encountered? There are many different kinds of stress. So the temperature is very common physical stress, but you can imagine other stresses, like, for example, oxidative stresses. So the production from the host of reactive oxygen species that can damage the bacteria, can damage DNA, can damage RNA in bacteria. So the bacteria can face to these stresses and can react to these stresses. Other conditions that are crucial for bacteria are conditions in which the nutrients are scarce, so conditions in which bacteria are starving. And they also need to cope with those conditions. Or different pH range, and they can also sense the presence of other bacteria or the presence of the same kind of bacteria species but a lot of cells surrounding the bacteria. So this is, they can sense the cellular crowding of the environment. And finally, especially for pathogenic bacteria, when they are in animals or in human, they need to face also another stress, that is antibiotic stress, because to fight against the infection, we use antibiotics, but bacteria has a different way to escape and to live in the presence of small concentration of antibiotics. Okay, so they can deal with these stresses using different strategies. So of course they can try to eliminate the stress. They can try to limit the effect of the stress. They can and they have to repair damages due to the stress. And finally, they can also try to escape to the stress. Okay, so how those bacteria are able to adapt to these different stress conditions? First of all, as I told you before, they need to understand that there is a problem, that there is a stress. Okay, there is a different environment, potentially harmful for them. And so for this reason, bacteria, they have evolved several two-component systems. There are very simple mechanisms in which we have a membrane protein that is just on the surface of the bacteria, so able really to perceive the stress very rapidly, and that in the presence of the input, so the stress in this condition, can be auto-activated by autophosphorylation on one histidine residues. This activated receptor of the stress will then activate a response regulator. This is why they are called two-component system, because it's made by two proteins. In bacteria, this is very simple. In eukaryotes, this kind of system are quite complicated with a chain of activation of different proteins until the effector. In bacteria, they are quite simple, just two proteins. And then the response regulator itself can do plenty of things. So it can activate transcription, it can regulate translation, it can activate other regulators, because in fact what is interesting it is that when you have a stress condition, the bacteria will not only do one thing, like for example counteracting the stress. They also need to repair the damages of the stress. They also need to modulate their metabolism in order to live in the presence of this stress. So all these things has to be done at the same time in the presence of the stress. So you don't need to just activate a few genes to do something, but at the same time you have to activate some genes, so you have to repress other genes, so you have to really create a network of regulation to cope with a single stress condition. Okay, so in my presentation today, we are going to see some of these two-component systems. In one case, this is a sensor domain that is not on the membrane. This is, we are going to see it is in Mycobacterium tuberculosis, and this is DOS-T protein is able to sense the absence of oxygen, so when the bacteria is in a hypoxic condition. And it is transferring the stress signal to a response regulator that is DOS-R. And we are going to see how RNA modification have an impact in this signal, and especially on the production of this DOS-R response regulator. Another system that we are going to see today, it is the AGR system of Staphylococcus aureus. So in this case, the system is meant to let the bacteria know that there are several Staphylococcus aureus cells that are in the same abscess. So as we are going to see later, this is a signal to tell stuff to change its lifestyle. There are too many cells in the same place. It's time to move and to colonize other organs to spread the infection. So there is a membrane protein, a GRC, and then it is activating a transcription regulator that is AGRA, that when it gets activated, is actually activating some gene, but is also activating some other regulators. In this case, RNA molecules that are themselves regulating the expression of several genes, repressing some gene and activating some other genes. But we are going to see in details later these two systems. So, as I told you, to respond to a single stress, bacteria has to put in place quite complicated regulatory networks. So this is just to give you an example. In this network that is in Staphylococcus aureus, there are several genes that are activated or repressing, producing virulence factors. So just keep in mind, today we are going to speak about pathogenic bacteria. Interaction with the host, it is done by several proteins that we name virulence factors because they are important for the virulence of the bacteria. So those virulence factors are actually regulated by the two-component systems, but also by several RNA regulatory molecules, so the small RNA, as we name it in bacteria, by other regulatory protein, transcription factor, translation regulators. So as you can see, it can get quite complicated. Okay, but today we will limit the discussion on the impact. I will give you example of how RNA modification can play an important role in these adaptative processes in pathogenic bacteria. So, as I told you, the different stress that we are going to look today are oxidative stress, cell density, antibiotic stresses, and those and other stresses can impact the level and the quality of the RNA modification in different RNA molecules. So the main molecules that can be modified differentially according to the stress are, of course, tRNA molecules, ribosomal RNA, but we also have regulatory RNA, the small RNA, and messenger RNA. So there is a main link between these molecules, that is the translation process. In fact, the majority of the regulation through RNA modification are at the level of translation. Okay, so what they can do, they can somehow perturbate this translation process. They can also allow translation in the presence of antibiotics, especially the modification of the ribosomal RNA. Those modifications can have an impact on the RNA stability, and so directly impacting, for example, the presence or absence of a regulatory RNA, or even recoding. So some modification on messenger RNA may change one codon for another codon, and thus at the level of the protein, we will have a different amino acid that will be incorporated into the protein, so we will have a different protein doing different things, or doing things more efficiently or less efficiently. Okay, so this is just an overview, just to tell you that RNA modification are dynamic. This means that we have some modifications that are constitutive, are always there, on the tRNA molecule, on the ribosomal RNA molecule, but they can also be modulated responding to this stress condition. So there are enzymes that can add the modification at specific position, and those enzymes may be more or less expressed in different conditions. But we can also imagine not only the involvement of enzyme for this modification, but just some condition may induce some RNA modification chemically without the need of a protein, an enzyme that will change the modification. So this is just to give you a perception of how many different modifications can respond to different stress conditions. And this is just from a review, a recent review from this year, just on tRNA molecules. Okay, we are speaking about modification. This week you had already at least 2 talks or more, I don't know exactly, but you should know that when we speak about RNA modification, we can have modifications that are very simple on RNA molecule, like methylation. So here we have a normal nucleotide, the adenosine, that is not modified. And then we have an example of a methylation added at position 1 of this adenosine. We can also have some deamination, like in the case of the production of the inosine, in which, in this case, we have removed the NH2 group from the adenosine. And those are simple modifications, okay? But we can have also much more complex modification, like, for example, the addition on a tRNA molecule on the anticodon at position 34 of the 5-methylaminomethyl 2 tau uridine. So from a uridine residues, we have added this complicated modification in 2 different atoms of the basis of the uridine basis. Okay, so for adding this complicated modification, we need many more than just one enzyme. In this case, for example, in this pathway in E. coli, there are 11 enzymes that are required to add this modification. So it is complicated, requires a lot of protein. Not only that, if you see in this pathway, I will not go in details of this pathway, but just to give you an idea of what we need to add this modification. We need a lot of protein, and we also need a lot of cofactors. So as you can see here, we need amino acid, cysteine, glycine, but we need also other cofactors, THF, FAD, GTP, SAM. So this means that in order to add this specific modification or other very complex modification, we need a huge pathway, huge apparatus, and a lot of energy. So there is a direct connection between the metabolism state of a cell, of a bacterial cell, and the ability to modify efficiently a specific nucleotide. So this is already telling you that the modifications are interesting as a regulatory hub, because they link directly a specific metabolic state with the function of the modification. Okay, so there are so many modifications, and the repertoire of modification is increasing day by day, not really day by day, but month by month. And there is this website you may already know, Modomics, that is collecting all the modifications that are newly discovered. And I took this screenshot this morning, so nowadays we have 172 different modifications that have been characterized on different RNA molecules and in different organisms. Okay, so what are doing this modification, why they are important, and why they can help bacteria to adapt to a different environment. So the modification can influence different edges of a nucleotide. So you know that RNA molecules have these 4 basic nucleotides, and they are able to interact with each other, forming RNA structure or RNA protein complexes by using different edges. So we have the classical Watson-Crick edge, in which you are familiar with when you have an helix that is formed or a base pair on the ribosome. You very often have this kind of base pair interaction, the Watson-Crick kind of interaction. Between, for example, the A and the U, or the G and the C. It is involving this edge. But we also have other interaction on RNA motif that also involve the sugar edge, this one, or the Hosting edge. Okay, and the modification that are here represented with the triangles here, they may have an impact on all three of these RNA edges, so producing a direct effect on the structure of the RNA molecule that has been modified. Because, for example, I will just give you some quick example. So we can have methylation of the ribose, not on the base, here on the ribose, on the 2' OH of the ribose, that can drive a specific Packer conformation of the ribose, that is the C3 endo conformation. So when the ribose it is in this conformation, it is more prone to give A-form helices. Okay, so the ribose in this Packer conformation, it is suitable to make a regular helix. Okay, so to have a stable structure with two strands that are interacting each other. Okay, and so we have a modification that is actually favoring this. But we have other modification, in this case on the base. So for example, the dehydrouridines that are actually doing the opposite effect. So these dehydrouridine are favoring the C2 endo conformation. So they are actually opposite to this. So they are, if we have an RNA molecule that has been modified with the dehydrouridine, you may not think to see a regular RNA helix that is formed, but rather they are present in loops and favoring this single-strand conformation. Okay, because they are also inhibiting an efficient base stacking. Okay, so already two examples to tell you that modification can have a direct impact on the structure of the RNA. Then we have also pseudouridine, and they are very dynamic. They can change their conformation very easily. And so they are present in RNA molecules that need to switch between different conformations, for example. At the bottom of this, it is that the modification can have an impact on the RNA structure, can have an impact on the ability of the RNA to be recognized by protein, we will see some example of it, on the stability of the RNA, and above all, on the decoding properties. So we are going to look more into the translation process, the decoding process, because it is there that tRNA modification and ribosomal RNA modification have a strong impact. Okay, so for that I choose to show you a movie. So please follow me in this journey into this movie, in which we are going to look at the ribosome, we are going to look at the decoding center, and we try to understand how it is formed, this decoding center, and how modification can change the ability to, for an anticodon to recognize efficiently a specific codon. I took this movie from Marat Yusupov group, and that has this crystallographer in Strasbourg that has do a lot of discovery on the decoding process on the ribosome, being able to crystallize both bacteria and eukaryotic ribosome. Okay, so let's see the movie together. We'll interact with the movie. So just okay. So we have seen this just to let you understand a bit more. This is the ribosome. Okay, it seems very complicated, but we are going to zoom in and to look only at some nucleotide of this translation machinery. So just to tell you, this is the large subunit on the top. This is the small subunit. And here you may see in a different representation, there are two tRNA molecules inside the ribosome. This is the E site tRNA, so it is a deacylated tRNA ready to leave the ribosome. This is the P site tRNA, in which we have attached the peptide that is forming into the exit tunnel of the 50S subunit. And then we are going to see in this movie how the A site tRNA will arrive on the ribosome and will establish codon-anticodon interaction into the A site that is here, that for the moment it is empty. Okay, from this view you don't see very well, but there is here on the small subunit an mRNA molecule already placed. Okay, so we zoom in. Okay, we zoom in and we start to see better the things. So here you see the mRNA molecule, you see the tRNA into the P site, and you start to see the ribosome environment. That is very important for the codon-anticodon interaction. So there are proteins. This one it is S12 of the small ribosomal subunit. There is, more than that, there are nucleotides from both the 16S RNA from the small subunit and the 23S RNA from the large subunit. So here in red we have an helix of the large subunit, that is helix 69, that is also participating in the decoding, in forming the decoding center of the ribosome, even if the majority of the residues are from the small subunit. Okay. Now we go inside, we start to look at some nucleotide. We can go ahead because, yes, okay. So this is not yet formed decoding site on the A site. It's not yet formed because you see here several nucleotides that are labeled. So we have nucleotide of the helix 44 of the 16S, A1493, A1492, and the 530G of the 530 loop. Okay? And then there is one nucleotide from the helix 69 of the large subunit, the A1913. So they are not yet in the right position. They will switch the position as soon as the tRNA will come into the A site. So this is the A site. It is still quite open, and it is ready to welcome a tRNA. But as you will see, very soon after the tRNA will come, it becomes very close. So become very close, and this is very important because it will impose specific geometry on the possibility to interact between the messenger RNA and the anticodon on the tRNA. And those geometry imposed by the ribosome are crucial for understanding how modification can have an impact on the decoding process. So for the moment it is quite loose, but we will see soon. So this is just a journey to let you appreciate that it's quite large at the moment, the A site. I will probably go faster ahead. Yes, okay. So here, okay. So here is the tRNA that arrived. Okay, so here just to let you understand, we have in red the messenger RNA and in yellow the A site tRNA that is coming. The conformation is still open, but very rapidly you see conformational changes on the ribosome. And as you can appreciate, in this region you have the contact between the tRNA and the messenger RNA, okay, between the codon and the anticodon. And all around this codon-anticodon interaction, there are all these residues from the ribosome. So now it becomes very close. Okay, and this is what I was telling you about the closing and the imposing of specific geometry. Okay, this is just to let you appreciate the conformational changes that are taking place. Okay, still from another view. The conformational change that will create the decoding center. Okay, and here again, you see it is very compact. Everything is very compact. So just a second. Okay, what do we see here? So we see residues of the ribosome, we see the decoding center, and then we start to see the interaction between the anticodon of the tRNA and the codon of the messenger RNA. So here we have the position of the codon. So in this case, we will have nucleotide 36, 35, and 34 on the tRNAs. So all the tRNA have the same numbering, even if they may change a bit the length, but by convention, we have the same numbering for all the tRNA. And the anticodon, it is always made by this nucleotide. So the 34, 35, and 36 in the anticodon loop of the tRNA. And as you can see here, there is, especially for the first two positions, only a single way to interact between the nucleotide of the anticodon and the nucleotide of the codon. Because of all these residues around that I named before are imposing Watson-Crick geometry. So you can have only Watson-Crick kind of interaction in the first and second position of a codon. While you see clearly here from these images that on the third position of the codon, corresponding to the interaction with the position 34, keep in mind, because several of the modifications we are going to look at are at the position 34 of the tRNA. In this case, it is much larger. There are no all these nucleotides around, and here we can have more freedom to establish different kind of interaction. For example, here we can have a wobble interaction between the uridine and the guanosine. Okay? Okay, so I think that I can go ahead. I will cut the movie here because this is what I wanted to tell you. That in the first two positions, you have imposed geometry Watson-Crick. In the third position, you may have also a wobble interaction between the uridine and the guanosine. Okay? So you can look at here to understand which are the differences between a Watson-Crick interaction, classical one, and the wobble interaction. You see here, this is linear. Okay? They are on the same plane, yes, but they are also collinear, the two nucleotides. In the wobble position, they are not linear anymore. You see? If you connect here in the middle, they are one above and the other below. Okay? What's going on when you have a modification? When you have a modification, you may change a bit this third position geometry. For example, here we have the classical wobble, UG, and here we have the same UG, but in the presence of a modification. So the uridine 5-oxacetic acid, this one, CMO5U34. And as you can see, what is changing here? You have a normal wobble, and here you have a UG that looks like having a Watson-Crick interaction. So a different one. When you have a Watson-Crick interaction, the stability it is higher because you have possibly one more hydrogen bond that is formed, and also because the stacking between position 34 and position 35 here, the stacking increase. So you see here they are not one above the other. Here you have a good stacking. So the result is that when you have a modification and you have this kind of geometry in the third position, you may have a higher stability of the interaction. How to explain this, the impact of this modification here to switch between this conformation and this conformation? Okay. In order to understand that, you need to know that, in fact, the uridine residues, they may exist in two different forms. The classical one, it is the keto form. But it is also possible to have an enol form. This one. In which, as you can see here, the hydrogen from the azote here has been given to the oxygen on the top. Okay, so here you have oxygen and then the hydrogen is here, and here the hydrogen is on the top. Okay, so naturally this uridine, it is most of the time in the keto form, and only a small fraction of the time will transit in the keto form. But it is possible that in the presence of the modification, there is a changing in the equilibrium between the keto and the enolic form of the uridine, thus favoring the formation of the enol form. Unfortunately, so far, we don't have the proof that this is the reason. We just can observe these differences in the decoding center, thanks to the work of Marathi Yusupov group and Venki Ramakrishnan group. For example, here the structure that I'm looking at are from Albert Weichselbauer. And they clearly show these nice differences. But the resolution of the structure is not enough to see hydrogens. Okay? So we can imagine that the reason why we have this Watson-Crick interaction between a modified U and a G, it is because of the U is in the enol form, but we don't have yet the proof. Okay, so now I will stop here this journey into the decoding, because we are going to see practical example of how to use this in the adaptation of pathogenic bacteria. Okay, you have listened in previous lecture different methods for detecting and characterize RNA modification in different molecules. The first modification that has been found on RNA molecule have been found already 50 years ago, and then with the development of new methods to detect modification, mass spectrometry methods or RNA-seq based method, RNA-seq based method. Nowadays we can detect modification not only on the tRNA or on the ribosomal tRNA, ribosomal RNA, but also on messenger RNA, also on regulatory RNA, so or on other RNA species. Okay, so this is why only recently we start to see the impact of this modification in RNA species different from the tRNA or the ribosomal RNA, because it is due to the advancement in the technology to detect those modifications. And we think that in the future we are going to see even more and more of this coming. Okay, so today I'm going to give you just a few examples of bacterial adaptation through modification in ribosomal RNA, tRNA, and regulatory RNA, and just one example of a modification on a messenger RNA, mostly in pathogenic bacteria. Okay, so I will start with a few examples of RNA modification on the ribosome. Okay, so this is the small subunit of the ribosome. From Staphylococcus aureus. As you can see, I placed here different modifications that actually we have found in our lab using different methods, mass spectrometry analysis, direct investigation of available cryo-electron microscopy structure, especially from Ada Ionat group, and in collaboration with Yuri Motorin, RNA-seq based method. Okay, so we have mapped several modifications in the Staphylococcus aureus ribosome, and here the numbering that you see on this modification are Staphylococcus aureus numbering. Because, for example, I'm going to speak about a specific nucleotide here that is demethylated, that is the 1411. This is the 1411 for Staphylococcus aureus, but this is actually the 1402 for E. coli. So if you look at the literature, you can find several papers on this nucleotide. In the majority of the cases, it is named 1402. Okay, but here I'm using the Staphylococcus aureus nomenclature. Okay, so what we know? So we know, for example, that if we delete the enzymes that are responsible for this demethylation, So there are two methyl groups that are added to this cytosine. So you will see that Staphylococcus aureus become more sensitive to the oxidative stress. So the oxidative stress is an interesting stress because the immune system, the cells of the immune system, macrophage, for example, neutrophils, they can phagocytate bacterial cells. This is one of the mechanisms of defense against pathogenic bacteria. And once they phagocytate the bacterial cells, they can stress them by producing reactive oxygen species. And they are creating an oxidative stress on the bacteria. An oxidative stress that can create damage on the DNA, can cleave RNA molecule, for example, and can also change the modification state of some RNA molecule. So the bacteria need to cope with this oxidative stress. And in this case, for example, we know that in the absence of these two methylases, so this nucleotide is not anymore demethylated, and then the bacteria is much more sensitive to this oxidative stress, so will be killed by the immune system very rapidly. Okay, and in this paper from 2015, they tried to understand a bit more why these modifications were so important in the oxidative stress. And then they figure out that this modification that is constitutive, so it is present on all the ribosome, Staphylococcus aureus ribosome, but also other bacterial ribosome, actually increase the fidelity of translation. So if you don't have this modification, especially under oxidative stress condition, you may form proteins that are aberrant proteins because you don't have anymore a proofreading mechanism that is efficient. So the amino acids that are incorporated into the protein may not be reflecting the real code that is present on the messenger RNA. So you may incorporate wrong amino acid. So you may have potentially killing effect of this protein that are formed aberrantly. Okay, so in this first example, we don't have really a mechanism, so we just have an observation of a modification that is constitutive. When it's not there, you have a bacteria that is not anymore able to produce efficient infection in the body. Okay, here I give you another example. In this case, we have antibiotic stress. Okay? So there are different antibiotics, for example, the erythromycin, that is a macrolide, but the same effect has been seen with other antibiotics like lincosamide or streptogramin B antibiotics. They are all binding in the same place of the ribosome. This is the exit tunnel of the peptide. Okay? So now you are a bit more familiar with ribosome, small subunit, large subunit, and then here you see the peptide in the tunnel. And here you see the binding site of the erythromycin antibiotic. Okay? So if we zoom in, we see that there are residues of some ribosomal protein that are surrounding this binding site, and there is especially one residue of this tunnel of the 23S RNA in this peptide tunnel, that is the A2085. Again, this is the nomenclature from Staphylococcus aureus. We are still looking at an example of a pathogenic bacteria, Staphylococcus aureus. In E. coli, the same nucleotide will be the A2058. Okay, so what we know it is that if we demethylate this nucleotide, we have resistance for these antibiotics. So the erythromycin and the other antibiotic, in the presence of a demethylated A2085 for Staphylococcus aureus, is not anymore able to bind. So we have a resistance against the antibiotic. Okay? But this resistant mechanism cannot be constitutive because it has been proven that those ribosomes that have this demethylation, they work less well than wild-type ribosomes that are not modified. So you need to modify the ribosome when the antibiotic is present, but otherwise you don't want to have this modification. Okay, there is a fitness cost for this modification. Okay, and this is very interesting because bacteria have evolved a very interesting system to deal with this problem. So they don't have the demethylation always present, but they allow the demethylation only when the antibiotic is present. In fact, the enzyme responsible for this demethylation, it is the RNA protein. And here you see the messenger RNA, the sequence of the messenger RNA for this RNA protein. As you can see here, in fact, this is the ribosome binding site, and this is the start of the decoding of the RNA. Okay? So the AUG, the start codon, it is hindered into a structure. This is the secondary structure of the messenger RNA. Okay, so usually this RNA is not produced. It is good because you don't want to demethylate ribosome in normal condition. Okay, this is silenced at the translational level because of this AUG and all this long helix that is formed around the ribosome binding site. But you see that at the 5' there is a leader peptide. So basically there is another sequence that can be translated by the ribosome. Okay? So if the ribosome translates this sequence, nothing happens, arrive to the end, to the stop codon, leave, and there is no perturbation of this structure. And this is what is going on in normal condition. But now we have the situation in which we have the antibiotics that is present. So the erythromycin is present. The ribosomes start to translate the leader peptide, but they are blocked by the erythromycin. Okay? This specific sequence is particularly sensible to the erythromycin action. Ribosomes are blocked here, so they are not coming to the end, they are not leading, they are favoring the formation of this alternative structure here. Okay, so they don't reach the stop codon, so this structure can be formed. This structure will abolish the formation of the inhibitory structure for the ERMC gene. This means that in the presence of the erythromycin, the second ribosome binding site on this messenger RNA will be available, and you will have the translation of the demethylase that will methylate ribosome and will give the resistant mechanism. Okay. Okay, so this just the two examples I wanted to give you for the ribosomal RNA, especially for the antibiotics resistant mechanism. And now we go with the tRNA modification. Okay, so this is the tRNA molecule. As you can see here, we have many more modifications that are present on the tRNA. Those are all the nucleotides that can be modified in the tRNA, and they, the modifications are accumulating in specific spots of the tRNA molecule. So in the anticodon, so those are the position 34, 35, and 36 that I tell you. As you can see here, 34, remember, into the third position, in the place in which the ribosome in the decoding center is more loose, can be very highly modified. So we have all these different modifications that can be found in different tRNA in position 34. Okay? And the nucleotides that can be modified in position 34 are uridine, but also guanosine, not only uridine. And then we have a position 35, not too many modifications, but there is anyhow some tRNA that have a pseudo-uridine in position 35. Position 36 is generally not modified, but in the anticodon loop we see other positions that are highly modified. Position 37, position 32. But then there are two other spots here. that are quite heavily modified in the tRNA. Those are the D loop and the T loop. Okay. So the modification here in the anticodon loop are really important for the decoding process. The modification on the D and T loop are important for the structure of the tRNA and for the interaction of the tRNA with the protein. For example, aminoacyl tRNA synthetases or modification enzymes, because there is a kinetics of the production of the modification on the tRNA molecule. And the first spots that are usually modified are the D and T loops. And then once they have been modified, you can have other modification enzyme that can recognize the tRNA molecule with D and T loop modified and can deliver modification on the anticodon loop. So these two spots are important for the structure, for the interaction with aminoacyl tRNA, with the modification enzyme, also with the EFTU. EFTU, it is the elongation factor that is important in translation to bring the tRNA to the ribosome. Okay. Okay, so now I will give you a few examples of differential modification on tRNA molecule that impact adaptation in pathogenic bacteria. And this is an example from Mycobacterium tuberculosis. Okay, so here you see Mycobacterium tuberculosis that is going to infect the primary site of infection. It is in the lung. And what is going on when you get infected by Mycobacterium tuberculosis? So first of all, there are some macrophages, as I told you, that are cells from the immune system that try to eat the bacteria. So they are phagocytating the bacteria. But for Mycobacterium tuberculosis, this macrophage action is not efficient. So in fact, Mycobacterium tuberculosis can still live inside macrophage. It is not killed by macrophage, and actually it is inducing enormous differences on the macrophage itself. So macrophages start to fuse together in the presence of Mycobacterium tuberculosis, and some granulomas start to be formed, in which we have a lot of macrophages fused together. So here you see the nuclei of the macrophages, several nuclei because they are fused in a single cell. And they start to recruit also T cells from the other cells of the immune system. And so they are organizing this huge mass of fused macrophage containing Mycobacterium tuberculosis cells and other T cells. And these masses, differently from a tumor mass, they are not served by blood vessels. So when you have a tumor, usually you also recruit blood vessels for the oxygen and to feed the cells. In this case, in the granulomas, there is no oxygen. There are no blood vessels that can reach, so there is no oxygen. So we are in hypoxic condition. Okay, so in this condition, Mycobacterium tuberculosis is sensing the low oxygen level, the hypoxia condition, and it is actually going into a latent infection state. So it decreases metabolism, starts to duplicate much slower. And in this condition, in which the bacteria is less active, is also more resistant toward antibiotics. So an antibiotic is very effective on a bacteria when the bacteria is in active duplication. When the metabolism is very active. Actually, more is active, more an antibiotic can kill the bacteria. But we can have quiescent bacteria that are not duplicating, that are doing nothing but surviving. And in that case, antibiotics are much less effective. So you can have a latent infection for many, many months because of the hypoxia created on the granulomas that is inducing a different lifestyle of the bacteria. So not anymore duplicating, not anymore doing whatever, but be there just surviving. Okay, and thanks to different publications here cited here, we are going to see how important is the modification on tRNA for this lifestyle change of Mycobacterium tuberculosis. In fact, they have done very elegant experiments to check the modification state on the tRNA under hypoxic condition. So here they take total tRNA from cells that were growing in normal condition in the presence of oxygen and cells that were grown under hypoxia condition, stronger and stronger hypoxic conditions. So here, for example, you have different days of growth in the absence of oxygen or in very limited amount of oxygen. And when they look at the presence of the modification on the total tRNA, they see that there were two modifications that were changing during this stress. So basically, they observed that a 5-methoxyuridine that was present before starting the induction of the hypoxic conditions decreased and disappeared, while another modification that was not present started to appear. Okay, that is the uridine 5-oxyacetic acid. This was the first experiment they have done. But in this case, they were looking at the total tRNA, so they don't know exactly on which tRNA species this modification is present and on which part of the tRNA. So they have done more experiments and they start to look more carefully into the treonyl tRNA. And in this case, they isolated treonyl tRNA and then they were treated the treonyl tRNA with nucleases, so to produce fragments that can be analyzed by mass spectrometry to determine the presence or absence of modification in the different part of the tRNA. And in this case, again, you have a normal normoxic condition and then different days after hypoxic stress. As you can see here at the beginning, the composition of this fragment here, including the anticodon, it was pretty much showing the presence of the methoxyuridine. Okay, so this treonyl tRNA, UGU, treonyl tRNA, in position 34 here is modified. The majority of these tRNA molecules are modified with the 5-methoxyuridine. Okay, but as soon as you start to have hypoxic condition, then the level of this methoxyuridine modified tRNA decreases, and what is increasing, it is the level of the uridine 5-oxyacetic acid. Okay, so you start with the tRNA that is normally modified, because the 5-methoxyuridine, it is what you expect to have as a modification on the treonyl tRNA in gram-positive bacteria, so Mycobacterium tuberculosis, it is a gram-positive bacteria with IgC content. And then, in hypoxic condition, you start to have a different modification on this tRNA. Not anymore the methoxyuridine, but the uridine 5-oxyacetic acid. That is a modification that is present on treonyl tRNA, but usually in gram-negative bacteria. Okay? Okay, so what's going on here? They went more into this phenomenon. They look at the proteome in hypoxic condition, and they see that there are some protein that are more abundant, but the majority of the protein is less abundant, and they look at the codon content of the protein that were more abundant. Okay, they were trying to understand whether this modification could have an impact only on specific messenger RNA because of those specific messenger RNA were containing some specific codons. So for the trionyl tRNA, for example, you can have 4 codons that can code for the threonine. Okay, so the 4 codons are the ACC, the ACG, the ACA, and the ACU. In normal condition, you have 3 tRNA species that are present: GGU anticodon, CGU, and the UGU codon that is modified. So in normal condition, you have that messenger RNA containing all these codons can be translated equally. But the majority of the messenger RNA contain ACC codon. Okay? Nevertheless, the 3 tRNA with the modified one that can recognize 3 different codons, the ACG, ACA, ACU, are equally translated in normal condition. But now let's see when we start to be in hypoxia stress condition. In this case, we have different things. First thing, the level of the tRNA GUG and CGU decrease. So you have much less of this tRNA. The level of the UGU tRNA increase a lot. And then, on the top of that, you change the modification, as we observed before. The result is that all those messenger RNA, the majority that were containing ACC codon, are not anymore translated because of the GGU tRNA, the only one that can decode this ACC codon, is not anymore there. And the other one, before they were translated equally with the modification 5-methoxyuridine, but now with a different modification, the ACA and ACU codon are much less well translated. And so the result is that you translate basically exclusively those messenger RNA that contain majority of the threonine codon ACG. Okay? Those messenger RNA include the messenger RNA for DOS-R. I don't know if you remember at the beginning I was telling you the 2-component system. So DOS-R, it is the responsive regulator for the hypoxia that is accumulating because of these differences in the tRNA modification state. Okay, so I give you more details about this because I found this very interesting because it is really linking the stress, the tRNA modification, the translation, and the outcome, the response to the stress condition. Okay, and this is the same that we have observed before, just to remind you that this modification, it is probably inducing this annual form, and so the ability, I just go back, the ability to translate the G here. The uridine modified, the uridine 5-oxacetic acid, can have a very stable interaction with the ACG codon. And we have here the explanation why, that I explained to you just before. What about the other codon? Okay, so we have that this modification can, probably in the annual form of the uridine, can have a kind of Watson-Crick interaction with the G, but in the same kind of geometry, the interaction with the A or with the U or with the C are much weaker because you have less hydrogen bond formation. So this is why the codon that is the preferred one in this condition, in presence of this modification, it is the G, the codon that terminates with the G. Okay. So now I will briefly give you other examples of modification in the tRNA that are occurring in specific stress conditions. In this case, we have Pseudomonas aeruginosa. There is another pathogenic bacteria that has Mycobacterium tuberculosis. It can be internalized by macrophages and in general by phagocytes. And as I told you before, this drive to the presence of an oxidative stress. Okay? Usually bacteria can die with oxidative stress, but Pseudomonas aeruginosa has developed defense mechanisms to cope with the oxidative stress. And those defense mechanisms require a specific modification on the tRNA in this variable loop. So we have an M7G in position 46. And what is interesting, it is that in the presence of an oxidative stress in Pseudomonas aeruginosa, you have increased level of this modification on the tRNAs. There are several tRNAs that can be modified in position 46. You have an increase in the level of this modification. But then, at the translational level, it is interesting to note that only few messenger RNA will get use of this increased modification to be better translated. Those messenger RNA are messenger RNA that contain a lot of phenylalanine and aspartic acid codons. And those messenger RNA are messenger RNA for protein that are catalases that can remove the reactive oxygen species from the cell. Okay, so in this case, we have oxidative stress inducing increasing of a modification on the tRNA with the effect to have a better translation of messenger RNA containing a lot of a specific type of amino acid. And those proteins that are formed can counteract the action of the oxidative stress. But we don't have the mechanism. So it is interesting, and I hope this is a very recent publication. I hope that we will learn more how this modification can induce specific translation of phenylalanine and aspartic acid containing messenger RNA. Okay, another example. In this case, it is in several gram-negative bacteria. So we know that one of the mechanisms with which bacteria can deal with the antibiotic stress, it is to remove the antibiotic. So there are efflux pumps that are on the membrane there to really pump out of the cell the antibiotic. So you don't have the effect of the antibiotic because you remove the antibiotic from the cell. Okay, thanks to this protein that are in the membrane of gram-negative bacteria. So here it is, this is a scheme for a wall of gram-negative bacteria with the two membranes here, the outer membrane, the inner membrane, and these proteins that are crossing the two membranes, the efflux pump. Okay, and there are other membrane proteins represented here. So what is interesting is that these membrane proteins, they contain a lot of proline residues. And has been also noticed that at the N-terminus of this membrane protein, including the efflux pump, there are pro-codon that are specifically of the kind CCC or CCU. The other possibility it is CCA and CCG. But in this membrane protein, at the N-terminus, we have an accumulation of CCC and CCU codon for the proline. And what has been shown, it is that, in fact, in this bacteria, this modification, methylation of the G37, so close to the anticodon here, G37 is necessary to translate those proteins. So if you remove the enzyme, TRIM-D is the name of the enzyme that is methylating the G in position 37. So if you remove the enzyme, you don't have any more this methylation, and the effect of the lack of this modification, it is that those proteins with the proline, a lot of proline, CCU and CCC codon, the ribosome stop there, and there is a +1 frame shift that is created. So basically, at the end, you don't have anymore these proteins that are formed. So you don't have anymore the ability to pump out the antibiotic from the cells. So the cells without this modification enzyme are more sensitive to the antibiotic. So again, we don't have mechanistic details differently from the other modification on the anticodon loop to explain why this modification can be specifically important for those CCC or CCU codon for the protein. This will be interesting to understand better, but what is clear it is that by targeting this enzyme, 3MD, we can have the possibility to sensitize the bacteria against antibiotics. So we can have a combination of an antibiotic treatment with another molecule that will inhibit the resistance due to the presence of the efflux pump for the antibiotic. Okay, then those are the few examples I wanted to give you for the tRNA. We rapidly go through the other RNA molecules that can be modified in pathogenic bacteria. So this is another class. It is the small regulatory RNA. The class is named small regulatory RNA, but as you will see, those are not very small in some cases, like the RNA3 molecule here, that it is more than 500 nucleotides, so not really small. And sometimes they are also named non-coding RNA, but also in this case, not all these regulatory RNA are non-coding. And this is again for the RNA3 an example, because in the RNA3 sequence there is also a part of the sequence that is actually translated into a peptide that is a toxin that is important for Staphylococcus aureus virulence. Okay, so we keep naming them small RNA, but they are RNA with regulatory function. Okay, so I show you just 3 examples of them. The RNA3 from Staphylococcus aureus, and then the tmRNA and the yRNA. Okay, let's start with the RNA3. Okay, in both here, across my presentation, you see I put the references for the work I was showing. Just to tell you that I put in bold here paper that are coming from our group, just for you. Okay, so this is a typical Staphylococcus aureus infection cycle. So Staphylococcus aureus, when it is in contact with the human host, the first thing you need to attach to the organ, to the tissue, in order to start making colonies that are at the beginning microcolony that are growing, protected by biofilms that is formed around. You see the colony is much bigger here, and at one point when there is a lot of cells here, then the bacteria, this is an abscess, then the bacteria start to remodel the biofilm, and the several cells can escape this abscess site, travel in the blood, and reach new infection sites. Okay, so you can imagine that different set of virulence factors are important in the different stage of Staphylococcus aureus infection. At the beginning, you need to have a protein for the adhesion to the tissue, so adhesin, protein that are membrane anchored protein, and that can allow the establishment of a colony on a site. Then you need to produce the biofilm. Then at one point you need to remodel the biofilm. You need to remove the adhesin so that the bacteria can disperse in the blood. And once in the blood, you need to produce a lot of virulence factor to protect the bacteria from the cells of the immune system. Because before they were protected by the biofilm. Now they are in the blood, in the presence of all the lymphocyte, macrophage, etc., etc. So in that case, during the traveling in the blood, Staphylococcus aureus is actually producing a lot of toxin and exoprotein with cytotoxic activity against the cells of the immune system. Okay, so you have adhesin, biofilm, toxin and exoprotein that are formed in different stages of Staphylococcus aureus infection. And one interesting thing it is that the switching between the production of the different virulence factors, it is mainly done by the quorum sensing system. So basically, when the bacteria is growing in small colony, it is still producing biofilm and adhesive. As soon as there are a lot of cells, Staphylococcus aureus is able to sense the crowd around him, a lot of other cells around him, surrounding him. This is the time to switch to inhibit the production of the adesin and the biofilm, and to start secreting toxin and exoprotein. So this is under the control of the quorum sensing system. And at the center of the quorum sensing system, it is this regulatory RNA molecule, the RNA3. Okay? As I told you at the beginning, this is a classical two-component system. So we have protein, AGRC, that is able to sense a peptide that is secreted, that is a thiolactone peptide. When there is a lot of this peptide, AGRC gets auto-activated, phosphorylated, and is able to activate a transcription factor, AGRA. What is doing AGRA? AGRA is inducing the transcription of the RNA3. And then it is the RNA3 that is orchestrating the network important to switch between an adhesion mode to a dispersion mode. Okay, so here the network is really complicated. Okay, I'm not drawing here and I'm not giving you details of this network. I give you an example of this network. Okay, so here we have this molecule that is actually repressing transcription factor, like ROT, activating the translation of toxin, like HLA, this is another toxin, or repressing the or repressing directly the translation of specific virulence factors that are on the membrane and are not needed anymore. Okay, how is doing that? This is a classical small RNA regulation mechanism in bacteria. So basically, the target it is usually the translation process. So in this case, it is activating HLA because of HLA has a structure in the messenger RNA that inhibits translation. But the RNA3 can bind to the 5' region of this messenger RNA, change the conformation of this messenger RNA, and allow the binding of the ribosome. Okay? So this is the activation mechanism. The repression mechanism, it is the opposite. So basically, the RNA3 molecule can bind to the Schindler gun of some messenger RNA, thus repressing the translation. This is a classical small regulatory mechanism of activation or repression of translation. Okay, so if we look at the RNA3 molecule, you see that the majority of the domain that are involved in regulation are at the 3' of this coding sequence. Coding sequence that is also coding for a toxin. So you produce the RNA molecule. RNA molecule is decreasing the adhesion, increasing the production of the toxin, using the 3' untranslated region of this messenger RNA. Itself, the RNA3 is also producing a toxin itself. Okay, so there is a combined action on the same molecule. Okay, so this is when there is a lot of cells, you start to produce the RNA3. The result is that the cell wall anchored protein, adesin, are repressed, and the exoprotein are expressed. Okay, and now we go to the modification, because very recently, this year, has been published by a group in Heidelberg, Andres Jaske group in Heidelberg. This is the publication. That the Staphylococcus aureus RNA3 molecule, it is modified. It is modified at the very 5' end of the molecule by a NAD molecule. Here. Okay. So they developed some technique to use RNA-seq to analyze the presence of this NAD modification at the 5' of different RNA molecule. And when they use this technique that is named NAD capture-seq in Staphylococcus aureus, they see that the main RNA that is modified by NAD at the 5', it is the RNA3. Okay, so now in other bacteria, in E. coli, for example, or in Bacillus subtilis, the modification of RNA molecule with the NAD at the 5', you want to ask a question? No? Okay. By NAD, the main effect, it is a stabilization of the molecules that are modified with the NAD. Okay, so in E. coli, we know that, for example, the main RNAs that is important for RNA degradation, it is the RNase E, and the RNase E need to have a substrate, an RNA molecule that is monophosphate at the 5'. So the presence of this NAD protects from the degradation by the RNase E in gram-negative bacteria. In gram-positive bacteria, like Bacillus subtilis, we have other enzymes that are 5' to 3' exonucleases that are the RNase J1 and J2. And so these enzymes, they are not able to destroy an RNA molecule if the molecule is protected by a NAD cap at the 5'. This is what we knew before. But they check the level of, okay, so I have to explain a bit more. How this modification it is added to the RNA. In fact, this modification is not added post-transcriptional. So we used to speak about post-transcriptional modification, okay? Because usually you have the transcription, and then you have another enzyme that is changing some of these nucleotides. This is not the case for the NAD molecule. In this case, it is a co-transcriptional modification. So basically, RNA polymerases can insert directly as a first nucleotide a modified nucleotide with a NAD. And this is what is interesting in this work, due to the sequence of the promoter. So depending on which kind of promoter, and actually which kind of nucleotide it is at the -1 of the transcription start site, you may or not incorporate NAD molecule as a first nucleotide. So this means that there is no enzyme responsible to add the NAD, but it is just the polymerase. This complicates things, because if you want to see the effect of a modification, you eliminate, you delete the enzyme responsible for the modification, and you look at the phenotype. In this case, there is no enzyme. Moreover, in gram-negative bacteria, we know that there is an enzyme that is removing the NAD. This is NUD-C. Okay? It is an enzyme that is not adding, but is actually able to remove. So, in gram-negative bacteria, you could do an experiment in which you delete this enzyme, so you have more NAD modification, and you compare with the wild-type condition in which this enzyme is present, and so you have less NAD modification. But in Staphylococcus aureus, there is no such enzyme. There is no NUD-C. So it is quite complicated to understand what is actually doing this modification on the RNA3. But they figure out that depending on the sequence of the promoter, they may have a more efficient incorporation of the NAD or a less efficient incorporation of the NAD. So they have been playing, changing the sequence of the promoter to increase the amount of NAD that is added to the RNA3, or to decrease the amount of NAD that is added to the RNA3 molecule. So this is what they have done. So basically, this is the bottom. They have been playing with these different promoter sequences. And the result, at the phenotypic level, it is quite controversial. So far, everything is very interesting. The outcome of this study, unfortunately, it is quite complicated. Because what they have seen, it is that when you increase the NAD level in the RNA3, you start to see that, for example, HLA is less induced. So normally, RNA3 induces HLA. With the NAD, it's less efficiently induced. Okay. But SPAR, for example, for the adhesion, SPAR messenger RNA that normally is repressed, in the presence of higher modification with NAD, is even more repressed. So on one side, we have a better activity of the RNA3 molecule. On the other side, a less good activity of the RNA3. They also compare the level of the RNA3 with higher modification or lower modification, and the level is the same. There is no effect on the stability. Or if there is an effect on the stability, it is counteracted by the more efficient transcription. At the end, you have the same level of the two, modified or not, RNA3. The production of HLD, so the translation of the RNA3, it is reduced in the presence of the NAD. Okay, so this is already quite complicated because it's not going all in the same direction. And things are even more complicated when they did the experiment looking at molecules that were containing less NAD modification level. Because in fact, for the regulatory function, it is the same effect. Increasing NAD or decreasing NAD, you will always result in less HLA activation. Increasing NAD or decreasing NAD is always result in higher SPA repression. And increasing NAD, you have less translation. Decreasing NAD, you have a translation that is similar to the wild type level. So this complicates a lot the outcome of this study. So it is interesting. It is an example of a modification on the small RNA, but yet the function of this modification is not yet clear and will necessitate much more deep analysis to be understood. Okay, now I will go quickly toward the end of my presentation, showing you very rapidly a sample of modification in other regulatory RNA. This is the tmRNA and the yRNA. So tmRNA is an RNA molecule that is made by a tRNA-like part and a messenger RNA-like part. You may know it already. What is interesting is that the tRNA-like part, it is modified. It is modified into this region that is really similar to a T-loop of a tRNA. In fact, we have the same kind of modification of a tRNA in the T-loop with the methylation and pseudouridine. Okay. This is the action of the tmRNA. So tmRNA, it is a rescue system. So when you have a messenger RNA that is truncated, does not contain anymore a stop codon, and in specific conditions like oxidative stress condition, you may have a lot of messenger RNA that are truncated because of the hydroxy radical that are produced in the oxidative stress condition or other reactive oxygen species that can induce cleavages on the RNA molecule. You may result in accumulating a lot of truncated messenger RNA. When the ribosome reaches the end of a truncated messenger RNA without a stop codon, it is blocked in this situation. So you have a peptide that is not a full protein and potentially could be harmful for the cell. And moreover, you have a lot of ribosomes that are blocked and that cannot contribute to the normal translation because the ribosome need to be recycled every time. To make the ribosome is a huge machinery. You need a lot of energy. You cannot make new ribosome to make new translation every time. You need to use the same ribosome for several messenger RNA over and over. So in order to remove this situation that is really harmful for the bacteria, there is this rescue system with the tmRNA. So the tmRNA is brought to the ribosome by EFTU because the tRNA-like structure will really resemble the tRNA, and it is also aminoacylated with an alanine residue. So EFTU recognize this as a tRNA, bring it to the ribosome. There is also another protein, SNPB, that is present here, and when the tmRNA interacts with the ribosome, you can have a peptide bond formation that is between the peptide that was already there, waiting, and the alanine and then you can keep translating the mRNA part of the tmRNA molecule. And so you rescue translation, and on the top of that you are adding a C-terminal peptide with a specific sequence that target the protein for degradation. So you remove a problem, two problems at the same time. You free the ribosomes, they are not anymore blocked, and you remove these aberrant proteins that are truncated protein by tagging them with this C-terminal sequence. Okay. So, but what is important, it is that the tmRNA has to be recognized by AFTU. So this is why the T loop has to be modified, because the AFTU is also sensible to the modification of the T loop to recognize tRNA and the tmRNA molecule. And this is important in adaptation and especially in oxidative stress condition. Okay. Now, there are a few studies that show that in pathogenic bacteria, like Salmonella species, if you suppress this tmRNA, you may have a 200-fold decrease in the virulence of this species. And more recent, it is already 20 years ago, and another paper could show, in fact, that this is because the tmRNA, SSRA, is the RNA part of the tmRNA rescue system, is affecting the expression of several virulence genes. Okay, we don't have details, again, mechanistic details. We just have that this molecule is important in adaptive condition, oxidative stress, is important for the virulence of pathogenic bacteria, Salmonella, and has modifications. Okay, so I found it interesting, even if there is no direct link and there are no mechanistic details, to see that in fact these modifications are important for the virulence of this gene. Here, just to show you the similarity between an AFTU tRNA complex and AFTU TM RNA complex. With the modification here, that are the one responsible for the recognition by EFTU. But there is another molecule that resembles the tRNA that is also modified and that has regulatory function. This is the Y-RNA. This one, you see, looks like the TM or the tRNA, and it is also modified in the T and D loop, similarly to a tRNA. What is doing this RNA? This RNA is important for the RNA homeostasis in the cell. So it is inducing degradation of specific target RNA because it is making a complex with a protein, RSR, and with the PNPase, that is an RNase that has a 3' to 5' exonucleotidic activity. Okay, so basically the function of this yRNA are to help this complex degrade the target RNA molecule. Okay. It is modified. The modification probably are there in order to produce this tRNA-like structure that probably prevent this RNA molecule to be degraded. So it is an RNA molecule that is in complex with the sort of degradosome, with the machinery to degrade RNA. But in itself, it is not degraded. So probably because it is stabilized by the fact that there has a tRNA-like structure stabilized by the presence of the modification. So again, also in this case, we just have a few evidences that show that if the pseudouridine modification on this yRNA is present, we have a virulent strain. But when this pseudouridine is not present, the strains are much less virulent. Again, this is just an observation. There are no mechanistic inside. We need to study it more carefully. But again, this is showing you how modification, RNA modification, may have a direct impact on the virulence of pathogenic bacteria. Okay, and then I finish really the very last example, showing you modification on messenger RNA. So messenger RNA are less abundant than ribosomal RNA or tRNA, less stable than ribosomal RNA, tRNA, or regulatory RNA, so they are more difficult to be studied. Look for modification and the effect of modification. This is why only recently we start to have example of modification of messenger RNA in bacteria. And one of the modifications that that has been found in messenger RNA, it is the editing. So the editing, I show you at the very beginning of my presentation, it is just a deamination of an adenosine that will lose, will be deaminated and will become an inosine that looks like a guanosine. Okay. So there is a modification enzyme responsible for this editing mechanism, that is the TABA enzyme. Okay, so this work from 2017 showed that in E. coli, so E. coli is not really pathogen, at least the strain that they were using in this work is not pathogenic, but nevertheless, they show that when you have high cell density, the STAT-A enzyme increase. So you have much more editing in the cell. But the editing is not all over. It is concentrated on specific messenger RNA, specific RNA molecule. And this is especially the messenger RNA coding for a toxin, that is HOCB. So when you have this change in the editing, the result is that you change a codon TAC to a TGC. Because as I told you, the deamination of the adenosine that will lead to the production of the inosine, it is actually similar to a guanosine. So will be read by the ribosome in the decoding center as a guanosine. So basically, this toxin changed tyrosine into cysteine. That become much more toxic. So before you have the production of this toxin that is not very active. Then, when you have high cell density, the enzyme change this tyrosine to the cysteine, and then the toxin becomes much more efficient. And the result is that you have a grow inhibition, because this is a toxin, autotoxin. Okay, so it is really affecting E. coli itself. But when you reduce the growth because of the presence of the toxin, the bacteria become resistant to antibiotics. So again, modification that induce antibiotic resistance. Okay, so we are at the end. I give you an example. You can find other examples in the literature, but you cannot find millions of examples because of the field is a new field of investigation, because it is still difficult, but we have now the means, and we can see that the field is moving forward with new methods that are continuously developed, especially to look at modification using RNA-seq methods and mass spectrometry analysis as well. So those methods allow really to look for specific modification, but on all the transcriptome of the bacterial cell, so also messenger RNA and other RNA species. And so most likely this field is in expansion, so we will see more and more examples of the impact of modification on the pathogenicity of bacteria, and how they are using this modification to adapt to the host. Okay, so here I give you some example of the expression of virulence gene to the response to the stress, the antibiotic resistance, but we will probably discover also other function of this modification in pathogenic bacteria. And then on the top of that, this is also inducing a new field because these modification enzymes may be the target of new antibiotics or new molecules that can cooperate with existing antibiotics. Okay, so just to briefly finish with the acknowledgement, so I will just show you, this is our laboratory in IBMC. So there are, as you see, several people. As I told you, we are studying Staphylococcus aureus. We are studying how Staphylococcus aureus adapt to the host, response to the stress at a different level, using a small regulatory RNA, using regulatory protein, etc., etc. And we are also studying the impact of modification in this process. So I am particularly interested in this branch of our study, the impact of modification, and I am studying it with basically two PhD students that I am directing. So this is Laura Antoine and Roberto Baena-Seron. So Laura is focusing on the modification of the tRNA in Staphylococcus aureus. And she has interesting but yet not published results showing how Staphylococcus aureus can adapt to the oxidative stress by inducing a differential modification of some tRNA. Roberto is actually focusing on the modification of the ribosomal RNA, and he is going really to the mechanistic details because he's also doing a cryo-electron microscopy analysis of ribosomes that are modified in a particular way under specific stress condition. And this is done also in collaboration with Bruno Klaus from IBMC in Strasbourg. And then also Hiroki Kazanawa, that is a postdoc working with me, is also somehow involved in these RNA modification studies because we found that in E. coli some tRNA that are not involved in the translation, but are actually involved in the formation of the peptidoglycan on Staphylococcus aureus, they are not modified. So this was really surprising. Now we are trying to understand the role of the absence of this modification. In fact, there are two machinery. One it is EFTU and the translation that has to recognize modified tRNA, and the other one it is this protein that are taking unmodified tRNA and are bringing amino-acylated but unmodified tRNA to the membrane and then to the peptidoglycan for making the pentaglycine bridge on the peptidoglycan of Staphylococcus aureus. So also here we have a role of RNA modification, not only in the translation, but also in the formation of the peptidoglycan. In this case, it is the absence of modification. And then the example I gave you, and many other examples we found in the literature, thanks to the work of three Master 1 students that been working with me and with Pascal Rombi, that is the leader of this group, during this summer. So because of the COVID, they could not come to have a stage in the lab. We decided to do a bibliographic work, and the final result, it is summarized in a review we are putting together to be published on RNA modification in pathogenic bacteria, impact on host adaptation and virulence. And they are Amy Dedi Bunwari, Martin Gobry, and Victor Logler. And so thank you for the attention.